Please do.
All right. Hello there. Welcome. Thank you so much for coming today. If you could introduce yourself with your names and the university and college you teach, and also give us an explanation of your field.
Hello, my name is Peter Hickmott, and I'm an associate professor in the Department of Psychology at the University of California, Riverside. And my primary research interest is really the basic understanding of the organization and plasticity of neural circuits. So the techniques I tend to use most are a variety of electrical recording, anatomical sorts of techniques. So I'm interested in circuits of all sorts.
Great. So how long have you been a professor?
Let's see, I've been at UCR now for 22 years now. Yeah, it'll be 22. Yeah, it's 22 years. It'll be 23. Yeah, so a while.
Yeah, I see. Wonderful. So do you have any like outside hobbies or interests?
I have many outside hobbies and interests. Um, I'm interested in history and I read a lot. I like to cook. I brew beer. I like to walk and hike. I like a lot of things.
I see. All right, great. So let's get into it. My first question to you is, what led you into your field of study, and where do you think it's headed towards in the future?
Well, so you could say if you had seen me when I was, say, 6 years old, you perhaps wouldn't be that startled as to where I ended up today. When I was young, I was interested in the natural world of animals and behavior. So, in fact, I can recall this is a good anecdote from a time when I was maybe 6 or 7, trying to mark ants with whiteout to better track their behavior and see what they were doing. So I was always interested, I guess you could say, in science and behavior. And so I really sort of had the idea that I might become, a behaviorist, a naturalist. When I went to college, when I took animal behavior and neuroscience, I became more interested in the genesis of behavior. By that, I mean the sorts of circuits and processes that underlie behavior, which led to neuroscience, rather than behavior for its own sake. So I guess you could say that's sort of how I ended up, where I am today. But it's would not have been a big surprise based on how I was when I was, say, 6.
I see. So, what questions are you answering if you did any research in the past or the future, and if you have any data or latest findings in your lab, maybe?
Well, I have lots of data. Remember, so for the UC system, faculty appointments, those are research appointments, not teaching appointments. So research is technically what I was hired for, not
Mhm.
teaching. Teaching is really secondary, but I do plenty of that. So as I say, I'm interested in circuit organization and plasticity. And that, of course, is an extremely basic general question. Obviously, all neural circuits are organized and they're all plastic. And by plastic, I just mean really capable of changing in meaningful ways. Again, that's a very broad statement. So in thinking about this, maybe the sort of easiest way to think about it is to think about your sense of touch. So if you touch a point on your hand, on your finger, a particular part of, say, your cortex will be active. And then if you touch an adjacent part will be active. So you can think of there as being a map or a representation of your body's surface at various points in your brain. Okay. So a question that I was interested in was, well, what is it about the actual circuit in the brain itself, the way the cells are connected together, how exactly they're connected, the strength of those connections? Why is it that the map is organized the way it is? Why is the transition between, say, the area associated with the forelimb, the forepaw, versus the, say, lower jaw, why is the transition point at a particular place rather than another spot? So how does the circuit organization inform the organization of the representation? Okay. And so in order to look at that, we developed a preparation a number of years ago looking at that question in rodents. So you can determine the location of that transition region between, say, the forepaw and the lower jaw representation, and then you can actually mark that region and analyze the cells and the circuits in that region to try and understand what's so interesting about that transition. And to sort of cut a number of years of research fairly quickly, basically there are a lot of things that inform that location, that transition. A lot of properties of the neurons, the ways they're connected, where do they send their connections, how those connections operate, their basic properties. And so, that was kind of interesting to see the reflection of the circuit in the representation. Okay. So that's all well and good, but it doesn't tell you anything about change, about plasticity. One of the nice things about this touch model is that you can look at change. As you may or may not know, if you lose a finger, for example, what happens to the representation of your hand in these areas of the brain that we're talking about? Well, they change. The area that is lost, say the finger, where there's no activity, the adjacent parts, the adjacent fingers in this case, basically expand into the area that's lost its activity. So the representation changes, okay, in a meaningful way based on the change in activity. Mm-hmm. Well, what about the circuit? We talked about all these properties of the circuit that tell you where that border should be, where it was. Do they change in parallel with this change in the organization of the representation? And basically, again, to cut a long story short, the short answer is that they do. All the different properties of the circuit can change in a sort of coordinated sequential fashion to, again, support and sustain this new organization. So again, you can really see the organization and change within the circuit itself as it informs the change in the representation. So that's, again, a very basic general thing that pretty much all areas of the brain do. That's how you learn things, right? You have changes in circuits that support changes in behavior. Mm-hmm. And so basically, we've looked at this in this touch model system a bit. I've also looked in a variety of other systems using different sorts of ways to cause change. And the short answer is that the sorts of changes are fairly similar between different types of change and in different areas. So the circuits change in similar ways in different areas to subserve these behaviorally relevant changes of various sorts. So that's sort of that's sort of the bulk of what I've been doing over the years and what we're still doing. I mean, we're still looking into these questions in various ways. Right now we're sort of better trying to understand the specific role of certain processes in these plasticity phenomena, these changes, these circuit changes. We're particularly interested in certain types of neurons. We're also interested in certain molecules that might serve to communicate changes in activity patterns, like when you lose your finger, that change in activity patterns. How does that change in activity inform all of these circuit properties? How does it inform cells? How does it inform connections? What sorts of molecules and processes might underlie that communication? And so we've been looking some at that as well. So that's basically, that's sort of the thumbnail 5-minute summary.
I see. So what are the ways like that neurons show plasticity?
Well, so a neuron has a great many ways that it can do that. Okay. When you think about, and actually, the neurons right behind, these are of course neurons right behind me, as you probably know. These are actually neurons in the hippocampus. And so as you can see, neurons have a very complex form. So this is a picture of what they look like. They have complex electrical properties and they have complex chemical properties. So all of these components, all of these pieces can change to inform the sorts of changes in say representation that we talked about. So you can see you probably know that neurons are connected by synapses, right? And synapses are ways that neurons use chemicals to communicate with each other. Synapses are very important for communication within circuits. Well, these synapses can change very rapidly. Okay. And synaptic change is one of the first things you see when you generate these sorts of changes in representation, for example. So changes in synapses, that's a big one. Okay. You can see changes in the sort of basic electrical properties of the neurons themselves. So the ways that the neurons handle their bioelectric events. You can see actual changes in structure. So neurons, of course, as you can see behind me, have very complex structures. They have a lot of basically input elements called dendrites. Mostly what you see here are dendrites. They also have outputs, axons. Both of those anatomical elements can also reorganize to subserve various sorts of change at the level of, say, the representation that we talked about. Okay, so there's a whole coordinated series of these sorts of changes that really affect all of this, the important properties of individual neurons as they are embedded in more complex circuits. Does that answer your question a little bit?
Yeah. So I was wondering, is there like a specific, let's say, equation to, I don't know, measure any activity of a neuron?
The short answer to that is not really. When you say activity of a neuron, that too is a difficult question to answer. Each individual neuron has, as you can see behind me, they have specific forms. So these are so-called pyramidal cells of the hippocampus. Okay, other cells look very, very different, for example. Individual neurons also have individual electrical properties. Okay, and that includes what you would think of as sort of basic cable properties that allow them to transmit electrical information along these long processes, for example, but also more interesting nonlinear active properties like action potentials. So these neurons, of course, use a particular bioelectric change, the action potential, to transmit information over their axons to their targets. Different neurons can have very different ways that they fire those action potentials, whether it's basically regular trains of action potentials or different patterns of activity. So different neurons have different firing patterns. There's no sort of single equation or that allows you to describe that we understand. It's based on the specific sorts of molecules that each of these neurons expresses, and of course where they're expressed and how they coordinate. So it's a very important property of neurons, this intrinsic firing pattern, and it is one of the ones that changes in the sorts of plasticity mechanisms that we were talking about. But there's no, you really, in order to understand that, you really have to measure it for a particular neuron. You have to go in and look. And that firing pattern is not, as I was just saying, it's not a constant. Neurons can change their firing patterns under different, say, behavioral circumstances or different brain states, like sleep, for example. So that firing pattern is really important, but also really dynamic.
I see. So 2 questions actually. So when you mentioned plasticity, is that like a broad term or are there different types of plasticity?
Plasticity is a very vague general term. It just means change. So the ability of, say, neurons to change in this case, or circuits to change, or behavior to change. So you can apply this term to any sort of change. You can talk about synaptic plasticity of synapses. You can talk about anatomical plasticity of connections. You can talk about molecular plasticity. You can talk about behavioral plasticity. So yeah, it's not a term that's it's very general. Yeah.
So I'm just trying to visualize how these circuits work. So does, like, when you say plasticity and let's say we're trying to learn something new, do the circuits in the brain expand like all around, or is there a specific region where this happens?
So the brain itself is made up of neurons, neurons and glia and other cells. And of course, those develop into specific circuits. As you probably know, the brain is not all uniform, right? It has different cells in different places. They're cooked together in different ways. So there are sort of functionally and behaviorally relevant regions within the brain that process different sorts of things. So you've probably heard of things like, visual areas or auditory areas or what have you. So not all the areas are equivalent. They're not all the same. They don't do the same things. So there are regions. Okay. Those regions are set up, their circuits are such that, the circuits are different. They have different cell types in them. They're hooked up differently. They may have different molecular, chemical messengers in them. So there are distinct differences that are, region specific within the brain, area specific. With that said, one of the things that the brain and the nervous system, one of the things they're really good at is change, learning. Okay, that's one of probably the most important things the nervous system can do, is learn new stuff. So in the face of new information or a new environment, generating behaviorally appropriate change in whatever behavior, internal state. And that requires change in circuits, just as we were talking about. And again, the ways that those circuits change at this very basic level, electrical properties, synaptic properties, connection properties, seems to be somewhat similar across regions. So the basic process that we're talking about in terms of circuits is a really global process across the brain to subserve this really important, behavioral process, learning. Every part of the brain basically learns. What exactly it learns depends on what it does. Does that make sense?
Yeah. So I'm just curious. So this kind of like a deep question. So when we learn something, right? When we learn from let's say some new information, we incorporate it into our brain, right? So how does that information travel and to a point where then it's then converted? I don't know. It could be converted into like a chemical message or electrical message, then connects to the circuits in the brain. Like, what is the whole process that happens? And then it's stored into memory. I think there's a whole process, right?
There's a whole bunch of processes. That's a big question. Yes, that's a big question that's not completely answered. Well, no, it is. I mean, there's a lot of people, a lot of labs, a lot of people working on that question, right? How do you learn something? How do you remember something? And that's again a very big question. The sorts of circuit level change that I'm talking about is clearly a part of that. In order to have a change in behavior, to learn something, things in your brain have to change, and they have to change over the appropriate time scale, whether you remember something for seconds or hours or days or weeks. There has to be some sort of mechanism that causes change within the appropriate circuit over that appropriate time scale. So again, as I sort of led off when I talked about my work, there's this coordinated set of changes in the circuit at the synaptic level, at the electrical properties level, at the connectivity level, at the molecular level. There are certainly ways that you can store information in the circuit then for extended periods of time, whether it's stable changes in connectivity patterns, whether it's stable changes in the expression of certain molecules. All of those things almost certainly happen in various ways. It may seem like I'm trying to avoid your question, but I'm
No,
not.
no.
The problem is it's as you say, it is a really big question. I mean, people spend their whole careers trying to understand little pieces of this. So to give that's sort of a reasonable 2-minute synopsis of a gigantic question.
Yeah. Okay. Well, here's my other question to you. So when you say plasticity, I know it varies between each person, right? So it's like, I just want to clarify. It's like the brain is a muscle, and as you learn more information, it strengthens, correct?
strengthens is a very, I don't know, loaded. It's a loaded word or an imprecise word. It changes,
Right.
certainly. So based on changes in the information that comes into the brain, whether that's caused behaviorally or, you know, by a lesion or what have you,
Mhm.
you said generate changes in circuits that generate changes in behavior. Um, Whether that's strengthening or not, I suppose it depends on how you want to define strengthening. It's change. It's meaningful change. So I guess you could think of that as strengthening.
Okay.
S- I mean, just to sort of pick a hole in that, if you talk about, we talked about changes in, connectivity between neurons, right? And that what that means is in some cases the strength of the connection between neurons gets stronger. In some cases it gets weaker. So some pathways are enhanced, some lines of communication are enhanced, and some are actually decreased or depressed. So there's again a coordinated sort of series of strengthening and weakening events, if you will, that are associated with these changes. It's not all about strengthening, I guess.
Right. So I just want to know, like, how do these like circuits like develop? I know, is there a process? I think I remember, is it called neurulation, something like that?
Okay, so you're also now again you're asking a gigantic question.
Okay.
How do circuits develop? Well, that's a very good question. So let me answer that in sort of two parts, because I'm not going to be able to answer it completely. Again, it's too big. The first point I want to make is that we're talking about these processes of change in, say, the adult brain, right? The way the brain is plastic. Well, it turns out the brain is also plastic during what you would think of as early development. How did these circuits get to be the way they are in the first place? One way they do it is, again, based on the activity, the information that they receive. That information acts on the circuit to pattern it in the first place. And then similar processes continue to occur across the lifespan to subserve things like learning. So what we've been talking about, this idea of plasticity and change, really also talks about development, at least
Mhm.
parts of development. Okay. So that's one part, just to relate it to what we've been talking about. And then there's a whole huge sort of other sets of developmental issues, some of which you were referring to. Obviously, we all start as a single cell. Fertilized egg is a single cell. Many things have to happen. We have to generate, hundreds of billions of more cells, not just neurons, but all the cells. So there are processes to determine what a cell becomes, what its identity is. Cells, they have to be born. They have to get to their correct locations by migration. Usually, when they get to their correct locations, they have to generate their correct, appearance. Like these cells, you have to develop all these very complicated connections. You have to develop your molecular and chemical identity. If you're a neuron, you have to generate your appropriate connections with your targets. So there's a whole huge process. What you're talking about as neurulation usually refers to the process by which, very early in development, you've probably heard of embryonic stem cells.
Mhm.
An embryonic stem cell is basically a cell very early in development. Those cells can take on any fate. So if you take a cell from that very early embryo, given the right sorts of, say, chemical or environmental stimuli, they can turn into any sort of cell: skin, teeth, bones, hair, nervous system. So they have any fate. What happens during development is those cells have their fates restricted. One of the first things that happens is those cells that can take on any fate have their fates restricted to, there are three groups of these cells, okay, that are going to turn into different parts, different cells in the body. The process of neurulation is basically the process by which a certain subset of these cells are instructed, you are going to become nervous system cells, neurons and glia primarily. That's usually what the term neurulation
Mhm.
is used for, that particular event whereby cells with many fates are instructed to take on just nervous system fate,
I
right,
see.
which is a fundamental event in nervous system development. Yes,
Okay,
but there's
so
all
they're
kinds of things that happen after that.
yeah, so they're like from a fertilized egg, and then they go through a process called embryogenesis, correct? Like
Yeah,
something.
embryogenesis. Yeah.
Cool. Yeah, that's so interesting. Well,
Yep.
so are there any like misconceptions in your field of study, would you say?
There are a great many misconceptions. What did you have in mind?
No, I mean, I'm just curious because we're in the field of science. I mean, a lot of people could disagree on like if we're doing research, especially some scientists could agree, disagree, but it is kind of a positive thing so that we have many different views on hypothesis, like you could say.
Well, so let's stick to sort of the topic at hand. You've
Mhm.
probably heard about or read about or seen various forms of, brain training games, right? These
Right,
are
right. Yeah. Huh.
apps or programs that are designed to train your brain to do something. Okay. And in some
Mhm.
cases, these training regimes make some pretty marvelous claims, I have to say.
Mhm.
So there's certainly debate within the field as to what are the limits of plasticity. What are the limits of these training programs? Because something that you see a lot with these sorts of training programs, let's say you're training some sort of, visual perceptual skill, so you train on that skill, the ability to detect, smaller and smaller differences in size, something like that. You do the training on the skill. You're going to get better at that particular task. Okay. You can train up almost any task. The big question, though, is does training on that particular skill transfer to something else that might be useful? So, for example, if you train your visual system on this particular task, does that, let you hit a baseball better, or improve your ability to see things when you drive, right? Because that's usually what these training programs are about. They're trying to use a specific set of basic task training to apply to something more behaviorally relevant in your life. And there's a lot more controversy. The evidence that you can transfer a specific form of training to a more global event or behavioral process, that's a lot more controversial, let's put it that way. And there's evidence in both directions, actually. It's clear that certain tasks do generalize to certain other tasks, but a task is not generalized to just any old thing. So there's one example of sort of an area of controversy in this particular part of the field.
Mhm. I
So
see. Yeah. I mean, I think I've seen quite a bunch of games of what you're talking about,
sure,
especially
there's whole
these.
there's whole there's whole businesses devoted to nothing but.
Yeah.
So, and I'm not going to comment on particular ones because I
Mhm.
don't really know.
I
But
see.
they do make, in some cases, very large claims,
Mhm.
which in at least some cases or many cases are not really backed up by any empirical science.
Mhm.
Then there are ones that are backed up with empirical science. So it's not that that approach is incorrect. It is correct, but
Mhm.
it's not infinitely powerful.
I see. All right. So I just wanted to ask, like, who or like what was your like inspiration to pursue your career, and any advice for students who want to be in a similar field to yours?
I don't think there was any single particular person except, that maybe my mom. She was trained as a biologist at university, and so she always encouraged us in our scientific endeavors. And my dad is a scientist too. I come from a scientific family,
Mm.
so I think that's probably certainly didn't hurt. I don't think there was any specific person along the way. I mean, and so no, not so much. Um, Advice for getting into the field. Well, I mean, first off, the field is a great field. Okay,
Mhm.
neuroscience is a great field to be in. It's a very dynamic field. It's a very diverse field. You can attack this question, as you may have gathered, from all kinds of different angles. There's all kinds of things that we don't know. So there's, again, there are wide areas of research that are still open, and so that, and you get to study the brain, which is very cool. So certainly I would encourage you or anybody else who's interested in the field to look into it. So research as a field is, it's a sort of a, I mean, research as a field in general, not just in neuroscience or biology, is an odd thing. It's really, it's really the last vestige of sort of the medieval guild system, where you're as a scientist, as a young scientist, you're trained by a mentor. So when you get your PhD, you're in a lab, and basically you're trained in that lab by that professor to do research. That's basically what a PhD amounts to. That's the bulk of your training, and you're trained to perform independent research. That's what your dissertation project is. It's a piece of novel research. So again, it's, you really, in order to succeed in research as sort of a career, in many ways, you really need to love research. So you need to love doing research and thinking about research, and you really have to value it for its own sake, because you're probably not going to make a great deal of money at it. A reasonable amount, but you're not going to make millions and millions. Not typically. That's rare. So, if you're interested in research, my best advice is try and do it. Get into situations where you can actually do research, whether it's independent novel research or lab classes or what have you. That's really going to be the most interesting and the most useful. Going forward in the career, it's important to think about research, the opportunities in research. And historically, you'd probably think about research opportunities, excuse me, as leading to, university professor, like me. That is a path. Okay? And it's quite a difficult path these days. There's a lot of competition for the positions. There are a lot of positions, certainly more than when I was applying back, 20 odd years ago, a lot of positions in industry, whether it's, biotech startups, drug companies, these sorts of things. So again, it's important to think about applying your research knowledge in different ways. Okay. Excuse me, sorry. So I guess that's, that's my advice.
Yeah, thank you.
Enjoy research. Enjoy research. Have fun with it. Research is fun. It's also hard work. It's, for stretches of time, you'll probably, eat, sleep, and drink research. And you got to sort of enjoy that.
I see. Yeah, that's great. I love that advice. Thank you. That sums up pretty much all my questions. Would you like to add anything last minute?
No. As I say, I think, neuroscience is a great field. Okay. It's not the only field, but it really is a very, a very dynamic field, a very interesting field. So of course you're interested in it. You should be. How can you not be interested in it?
Yeah.
So I mean, it's been a good career for me. I love research. It's what I do best. I enjoy the teaching, and I enjoy mentoring students. I like talking to students like you.
I see. I can tell. Yeah. Thank you so much for coming today. Taking time out of your day. I really appreciate it, and I hope you have a great day.
You too. Thanks for having me over and having a chat. If you want to have me talk anymore, you can always email me. So if you have questions.
Thank you. Bye.
You're welcome. Bye. Good luck.